Okay, well, I am going to review the topic of food dyes and children's behavior. We talked a bit about this yesterday, about defining the terms that we're going to use, and I'm going to be referring, when I'm referring to my own research, to hyperactivity. Hyperactivity is a pattern of behavior showing marked individual differences in the general population, and that's the key thing that I want to argue for, is that I'm talking about individual differences in the general population. It comprises overactivity, impulsivity, and inattention. Children at the extreme end of that range of hyperactivity may get diagnosed as ADHD with certain conditions that apply. And I'm going to argue later in the talk that increased hyperactivity is associated with later behavioral difficulties and antisocial behavior and educational problems. Just to emphasize that point, I'm talking about an aspect of behavior with that marked individual differences showing a roughly normal distribution. And if you're at that far end, then you might get diagnosed with ADHD. And I'm just going to try and get this. And the point is, I'm talking about variation along this dimension and what's the significance of that. This notion of rethinking the whole topic of ADHD as a dimension has been adopted by a number of people, including pediatricians. This was a quote from one of the editors of JAMA Pediatrics. We should move away from a diagnosis of ADHD to one of attention hyperactivity spectrum disorder. Accordingly, the shift will be treating attentional capacity from treating attentional capacity as a clinical disease to recognize that we need to do all we can to help children maximize their ability. And that's the theme of what I'm going to be arguing for. Now, later in the morning, I hope he comes along. Joel Nig will be here. And this is just a summary of some of the causes of ADHD that he has summarized. Very importantly, genetic differences, I'll come back to that in a second, are a major determinant of the condition. But of course, we're interested in environmental effects. And there are a number of what he labels uncommon experiential risks, prenatal alcohol exposure, nicotine exposure, although actually, interestingly, just this year, Rutter and Thapper have re-examined the notion of prenatal nicotine exposure and showed that that's probably not causally related to ADHD. It's more accounted for by genetic background. We've known for a long time that low birth weight is a risk factor for ADHD and high-level lead exposure. I'm going to come back to lead at the end of the talk. Lastly, what we're interested in are probably common experiential risks, such as diet. So genes are important. How important? Well, there's been a lot of research. I've done twin studies on this topic myself over the years. And just recently, Stephen Hinshaw has summarized the data that we have available and suggests that over 70%, 0.75, of the factors contributing to the risk of ADHD stem from genetic differences between children. That's basically a behavioral genetic approach, quantitative behavioral genetic approach. More recently, we've been doing molecular genetic studies, and a paper that's just recently been published in Nature Genetics comes up with the following conclusion. They've identified 12 genome-wide significant loci associated with ADHD. And the point that I want to make here is that they've emphasized that the results highlight strong overlap with the genetics of ADHD-related traits and health risk behaviors in the general population, encouraging a dimensional view of ADHD at the extreme end of a continuum of symptoms. So even from a genetic perspective, we're looking to try to understand this as an extreme on a continuum. So, hyperactivity is caused by a wide range of contributing factors acting in concert to increase the risk of hyperactivity in the child. We're interested, or you're interested, in food colors. Now, there have been a number of meta-analyses, one of which was referred to yesterday by Sharp and Trin, that have looked at the studies that have been done on colours and children's behaviour, and ADHD in particular. The first of this was the Sharp and Trin meta-analysis, and this looked at all RCT studies, randomized controlled studies that have been conducted up to that time in 2004. And they estimated an effect size of 0.28. The effect size here means that the exposure to food colours was shifting the scores of these children by about a third of a standard deviation. Joel Nigg, who's I think coming along later this morning, has also done some very sophisticated meta-analyses. His conclusion was that if you use parent ratings, you get an effect size of about 0.18. If you use teacher and observer measures, it goes up to 0.22, and then here he is. And then if you look at attention measures, the effect sizes go up even further to about 0.28. We did a meta-analysis, but we were concentrating on children with diagnosed ADHD. Many of these other studies didn't have a diagnosis of ADHD, they simply had elevated scores on ADHD measures. When you do this more restricted analysis, we found an effect size of about 0.42. So you can see there have been a number of people looking at this literature coming up with the conclusion that as far as children with elevated scores concerned, particularly with ADHD, there is an effect of artificial food colour on behaviour. But most of these studies were done some time ago. Most of these studies concern children with ADHD or high levels. The samples are often very small. There was none over 50, and most were under 25 children. Joel Nigg will be reviewing these in his next presentation. That raises the question, what's the possible effect of colours on behaviour in children in the general population? Because those studies are all about high schoolers, children that may be particularly vulnerable. That's where our work comes in. It's referred to as the Southampton study repeatedly, but actually it was two studies. One done on the Isle of Wight, and they're really annoyed that it's only the Southampton one that gets referred to, because they're a very proud island just off the south coast of England, and they want recognition too. The first study was done there. Yeah, it was published in the Archives of Studies of Childhood. Both the Isle of Wight study and the Southampton study were randomized, double-blind, placebo-controlled crossover trials. A pretty tight experimental design, and indeed I would label it as the gold standard for establishing causality in this particular field. We had a large sample. These were 3-year-olds in the general population, nearly 300 of them. They were screened before the study started for high levels of hyperactivity and for atopy, allergic sensitivity. We used skin prick testing to see whether they were reacting to allergens. We took a baseline assessment of their behavior, then additives were withdrawn from the diet. Measures of them were taken before and after the placebo and active challenges. I'll go into these in a bit more detail when I come on to the Southampton study. In this study, of the 3-year-olds in the Isle of Wight, we used parental ratings as our outcome measure, and we also attempted to use tests in the clinic, which was really quite a challenge with 3-year-old children. It was really quite difficult to get those done reliably. I'll talk about the challenges that we used when I get on to describe the Southampton study. What I want to emphasize here is that it's a large-scale study. It's nearly 300 children, and these are children in the general population. They haven't been referred, and their parents were prepared to put the children onto a modified diet over a 6-week period while we did our study. So it's really a very challenging thing to have done, and I think our research team did really well to carry it through. What were the findings? If you look at the change pre-placebo to post-placebo, the change in hyperactivity level scores was about 0.38. This is on parent ratings, by the way. There was a bigger change before and after exposure to the active challenge, 0.77. If you do the calculation down here, the baseline standard deviation was 0.76. You put that in the denominator of that equation, look at the difference in the means, and you get an effect size of 0.51. Quite substantial. Now, clearly not all children were reacting to the same extent. We were interested in, do the children that react more than others have distinctive characteristics? If you remember, we looked at skin prick testing because we thought maybe this was an allergic reaction that was determining the response. And neither the presence of atopy or indeed the pre-study level of hyperactivity were related to the size of the change that we saw. The most parsimonious explanation is that the extreme scorers were just on the end of a continuum. This study had been funded by the British government, and by the time we finished it, the Food Standards Agency had come into operation, and they decided that they wanted this to be replicated, which is why we went on to the mainland and did the Southampton study. This was published in The Lancet in 2007. The design, as I said, was basically the same as we'd done on the Isle of Wight. So one element of this was simply a replication with 3-year-old children of what we'd done before. But now we added a sample of older children, 8 and 9-year-olds. And you can see that, again, we've got quite large sample sizes here, 153, 144. And again, they were children from the general population. They weren't referred cases. They were just from local schools and preschools. We had children on a baseline measurement, then usual diet, and then with the usual diet, and then additives were withdrawn. And over the subsequent 6-week period, they were exposed either to a placebo or a mix of food additives. Hyperactivity we measured, this variation, this continuous variation, using parent ratings, teacher ratings. We put our research staff into schools and actually did direct observations of behavior in a school setting, using an Abacoff well-developed system for measuring hyperactivity. And we also, for the older children, for 8 and 9-year-olds, used the continuous performance task, which is a measure of attention that we can use for children. We did an attempt to use the laboratory tests on the 3-year-olds again, because they hadn't worked in the previous study. Now, these 3 sources of information, parent ratings, teacher ratings, observations, and for the older ones, the computerized tests of attention, were aggregated together to give an overall index of what we call the global hyperactivity aggregate. People are criticized us for doing that, but I actually think it's the most appropriate thing to do. It's more likely to give you a reliable effect if you've got multiple sources of information than if you would just take them in isolation. And when we designed our study, it was powered to detect an effect with this as our primary outcome. Right, let me give you a bit more detail about what we did. The children were studied over this 7-week period. First of all on their normal diet, then artificial food colors and preservatives were removed, and they were put onto a placebo drink. I'll come back to these in a second. They then went through a series, either active and placebo challenges, a washout period, active or placebo, washout, active and placebo. They were tested weekly on all the measures I just described to you. And they were randomly allocated to one of these 6 sequences of placebo, mix B, mix A, mix P, mix placebo, and B. I'll explain the mixes in a second. And it was a crossover trial, so each child was receiving each of these exposures. And so in that sense, they were acting as their own control. These were the challenges that we used. These are the names that we've been hearing about yesterday. These are the European E numbers, and these are the names. This was the mix A, had tartrazine, sunset yellow, carmazine, ponzo 4R, and sodium benzoate. I'll come back to that in a second. That was the mix that we used on the Isle of Wight. So for the 3-year-olds, that was a straight replication of that challenge. We also, for both 3-year-olds and 8-year-olds, had the second mix, which had quinoline yellow, sunset yellow, carmazine, and allura red for allura red AC. And again, sodium benzoate. Again, there's been some commentary about whether the exposures that we were using here were too high or too low or appropriate. I've labeled here, in this brackets here, the Dole figures for what were the 90% percentiles for 2-year-olds plus on the 10 to 14-day consumption. And you can see that our exposures for the 3-year-olds, and we had slightly higher exposures for the 8-year-olds, are of an order of magnitude similar to the ones that are being reported in the American population. Mix A was the same as the challenge that we used in the Isle of Wight. Mix B was the levels were increased somewhat and changed somewhat to reflect what the Food Standards Agency told us was the more average daily consumption of 8 and 9-year-olds in the UK. Oh, damn it, I think the system here has lost something for me. What I was going to do was to point out to you that although we are not just using the colours that are recognised in the States, the key ones here that were commonly used in the Dole study were also included in the challenges that we provided. So, what were the results? I'm going to present the results for 3-year-olds and for 8-year-olds. We presented the analyses in a number of different ways. Here are the mean scores on Mix A, Mix B, and on placebo for 3-year-olds. And you can see that they're both, on the mixes, their scores are higher than on placebo. Over here, we test whether that's significant or not. And if there's no significant difference, then that dotted line goes there, and the confidence in 95% confidence intervals will cut it. So you can see that what we've done here is to replicate what we did in the Isle of Wight. For 3-year-olds, Mix A was showing a significantly higher level of hyperactivity when the children were exposed to Mix A than when on placebo. Mix B was showing a strong general trend in the same direction, but it just failed to reach significance. Now, people have been commenting about our results and saying there were differences between the challenges and whether the effects were shown and the effects were not. That kind of view of it is being driven by p-values. And we know that you can just flip either side of a p-value and get a significant effect on that. What I think is much more important is to concentrate on the effect size and look at the pattern of the effect size. And you can see the most obvious interpretation of this is that for 3-year-olds, the effects of Mix A and Mix B are roughly similar. They're both around about the 0.25 level. This then presents results for 8, 9-year-olds. Again, you can see that the scores, the general hyperactivity index scores are higher when they were exposed to Mix A and Mix B than when they were on placebo. Oh, I didn't explain about the placebo, did I? What that was? No. That was a matrix made up of fruit juices largely, and some other contents that our nutritionists had developed, like beetroot and so on. It provided a color mix, which when you added our colors to it, couldn't be detected as being different. And so the placebo challenge was constant through both the placebo, the Mix A and Mix B. It's just that when they got Mix A and Mix B, the colors were present. We tested whether anybody could tell the difference between these, and nobody could, or not reliably so, anyway. What are the possible biological mechanisms? If I'm arguing that food colors are having an impact on hyperactivity, how does that come about? Back in the early 90s, I think it's in the 90s, I can't remember, 1990, there were a number of studies that were done that were suggesting that maybe food additive intolerance was mediated not by an allergic reaction, but by a pharmacological release of histamine. that this was a pharmacological release of histamine, and that was producing the effect. We don't think it was mediated by an allergic reaction, because if you remember on the Isle of Wight, when we looked at atopy, atopic children were not more likely to show a response than others. So we think it's possibly mediated by a non-IgE mediated histamine release. That's what we proposed in the Isle of Wight study. Now, for the Southampton study, we also took DNA from the children. And we're interested in whether there could be genetic differences between children that would explain differential vulnerability to the exposure to additives. We chose some genes that we knew were involved in the histamine system because of that prior prediction that that was the mechanism. But also it seemed to make sense to take genes which at that time were thought to be important in relation to ADHD and hyperactivity. So we used histamine N-methyltransferase polymorphisms, and also in dopamine system. And what we found was that the adverse effects of additives were affected in 3-year-olds and 9-year-olds by polymorphisms in the histamine N-methyltransferase gene. There were other genes that we looked at, particularly those related to ADHD, which didn't show an effect. So what conclusions do we get? From these two studies, Southampton being a pair of studies, so 3 actually altogether, large studies, mixtures of certain artificial colors together with sodium benzoate preservative in the diet increase hyperactivity in 3 and 8 and 9-year-old children in the general population. The average effect of these of additives on these general population samples is similar to that found by Nig and his hyperactivities, where most of those studies are on children with high levels of hyperactivity or ADHD. So although the results suggest that some mixtures of certain artificial food colors and benzoate preservatives may affect the level of hyperactivity for children in the general population, removal of these from the diet will not be a panacea for ADHD. ADHD is caused by a whole variety of different factors. One of the study nutritionists was Chris Locke. She went back to Hong Kong and did a study, not exactly replicating, but extending the work. She did a study where she separated the food colors and sodium benzoate into separate challenges. These were the mixes that she used. She only had ratings, no classroom observations, and she was studying 8 and 9-year-olds, and so importantly, she had no computer test retention. She concludes that the removal of the additives, she had a similar design, removal of the additives did lower the level of hyperactivity in these children, but putting them back in did not return the levels of hyperactivity back to their pre-trial level. This study is very difficult to appraise and interpret. The presentation results don't give you the actual means of standard deviations pre and post test, and the presentation of the linear mixed modeling is less than adequate. So it's difficult to appraise. No, John may say something about this later. Now, that's what I think we found. I want to now put those findings into some context. We used mixture of additives. Why did we use preservatives, sodium benzoate? In one of the studies that got mentioned yesterday on the withdrawal diet work, Egger et al. Found that both colorants, particularly tartrazine, and preservatives, particularly benzoates, were the common, most provoking substances for abnormal behavior in their studies. That's why the British government wanted us to put the sodium benzoate in. So we use mixtures, but colors are not used in isolation, which I think is very, very misleading. All this work that's been done on single isolated colors doesn't take into account the possibility that there are interactions between the colors and their joint effects. it was mentioned yesterday, that's a complicated issue. Well, it's complicated and it's also expensive. Can you guess how much our work cost, the Southampton study? It's going on for $1 million. That was for a mix. Now, if you were to replicate, and you need that kind of power, you need that kind of sophistication and measurement to get out these effects. If you replicated that for every individual colour in isolation and ignore the effects of mixes, you've got a huge financial cost. The effects weren't seen for all children. No, some children responded more, some children responded less. But there was no simple indicator. We looked at gender, we looked at social class background, we looked at A to P hyperactivity, as we said. There's nothing that we could find that indicated vulnerability, except for the genetics. Now, I'm sure you're not going to go around and propose that you screen your populations for genetic vulnerability to additives and then just take it out for those children that have that kind of genetic vulnerability. Not all the colours, oh, sorry, I thought I'd lost my slide. Not all the colours were FDA approved. This was a bit that I thought I'd lost. The colours that we included in our dyes, red 40, yellow 5, yellow 6, were the ones in the Dole studies that were the most commonly used, exposed, and taken by children. So I think there's a connection, there's a relevance. And I now want to move on to how important these effects are. Do ADHD symptoms in young children matter? Perhaps let's grow out of it. They don't seem to. I can give you results from some studies that have done follow-ups of children identified with ADHD in the young population. This was done by Lee in the States, and they're suggesting that very few young children diagnosed with ADHD are well adjusted over a period in mid-adolescence. Very few are well adjusted. There's a continuing risk. It's quite clear that the problems of inattention, hyperactivity, and impulsivity, those factors that are in our hyperactivity measure, are correlated with a host of achievement problems in elementary school. This was Spira and Fisher in multiple countries in 2005. So having a diagnosis of ADHD early on seems to predict something about how you're going to develop later. What about the individual differences? That's what I was interested in the general population. Does it matter whether you're a little bit higher or a little bit lower on hyperactivity than other children in the general population? But it seems that it does. This was a classic study from the groups that are doing longitudinal studies in New Zealand, by McGee et al., that there was a strong linear relationship between early hyperactivity and later adverse outcomes. Adjusted for other variables and so on, early hyperactivity was associated with continuing school difficulties with attention, poor reading, and adolescence. A linear effect going across this dimension. A more recent study that's come out from England, at a population level, after adjusting for confounders, there's a linear association between each 1 point increase in attention symptoms and worse academic outcomes. That was looking at an important end of school examination in the British system. So individual differences in hyperactivity do matter. Is an effect size of 0.2, which is roughly what we've been talking about, does that matter? It seems very small, doesn't it? It seems tiny. Let's put it in a bit of context. Using meta-analyses by Schwartz and by Pocock, the group at this in Georgia looked at lead in children. And what they calculated was that by falling blood lead levels were associated with a change in IQ points of about 0.2, 0.47 in the US population. That equates to an effect size of 0.23. If you take the difference between the average of those two values of the IQ points, you get 0.35. Standard deviation of an IQ is 15. That produces an effect size of 0.23. So in the general population, the effect of food colour on behaviour is roughly similar to that of lead on IQ. That makes this figure of 0.2 quite important. I think people thought that lead was quite important. It's important we took it out. It seems to me that in the general population, colours are having a similar effect size. I'm confused about time. Am I right with... take that as a yes. I'll carry on. What were the clinical implications of our findings? Paediatricians were reassured that they should take the findings, the reports from parents, seriously about the effect of food on behaviour. The impact in the UK and the EU, if you remember, we were funded by the Food Standards Agency. And in response to our work, because they couldn't act independently, food colours are regulated across Europe-wide, so they could only ask for a voluntary action down here, asking manufacturers to remove these artificial colours by the end of 2009. The European Food Safety Authority, they reviewed our work and they didn't recommend any changes within the European system, although the European government did. They said, well, this is the European Union government response, based upon these new data, they now require food labels across Europe, and that includes the UK, at least until we have Brexit, which may put us outside Europe in a few months' time. We have to have this label, may have adverse effects on activity and attention in children if it contains any of the food colours in the Southampton study. What has happened in the UK? There was a review here by Saltmarsh. Specifically, the colours used in the Southampton study, or the impact of food colours on hyperactivity in children, have been largely replaced by colouring foodstuff, and the preservative used in the study, sodium benzoate, has been replaced by potassium sorbate in the majority of drinks. So the situation in the UK has changed quite dramatically. Just to finally recognise the colleagues that worked on the study with me, John Moore is a paediatrician and immunologist, Edmund Smith, the Bartonella psychologist, John McCann, who was the first author on the paper, our dietitians, and Debbie, our secretary, and our various research assistants that did the observations. Thanks very much.